Good morning, everyone, and thank you for being here today. I am delighted to welcome you to this presentation. Today, we will be discussing the qualification of working standards against reference standards. My name is Dr. S.K. Singh, and I hold a PhD in this field. Over the course of this presentation, I will walk you through the key concepts, the importance of these standards, and how we ensure their accuracy and reliability. So, let's get started and explore this important topic together. Let's start by understanding what a working standard, or WS, actually is. It's a secondary standard that has been qualified against a reference standard, or RS. These WSs are prepared either in-house or procured from external sources, depending on the need. They are primarily used for routine analysis, batch release testing, and stability testing to ensure consistent product quality. It's important to note that WS must be qualified and controlled internally to maintain their reliability. Now, shifting to the reference standard, or RS, these are highly purified, well-characterized materials that serve as the benchmark for our measurements. They are used for critical activities like method validation, calibration, and testing for impurities and assays. Typically, RS are obtained from recognized pharmacopoeias such as USB, EP, BP, or IP. Because of their high purity and rigorous characterization, RS are considered primary standards with the highest level of accuracy. The main objective here is to establish a clear procedure for qualifying working standards to ensure they are traceable to the reference standards. This traceability is crucial for maintaining compliance with regulatory expectations like GMP, ICH, USB, EP, and IP. Ultimately, this process helps us uphold the accuracy, reliability, and integrity of our data, which are essential for product quality and regulatory approval. When it comes to key regulatory expectations for working solutions, it's essential that our qualification process clearly demonstrates several critical aspects. First, we need to verify the identity of the solution to ensure we're working with the correct substance. Next, purity is crucial, confirming that the solution is free from contaminants or unwanted impurities. We also need to assess its assay and potency, which tells us how much active ingredient is present and whether it's within the expected range. All these assessments must be performed using validated analytical methods. This validation is fundamental because it guarantees accuracy, reliability, and reproducibility of our results. Moving forward, maintaining proper documentation is vital. This includes keeping comprehensive certificates of analysis, or COAs, and establishing a traceability chain that links each batch back to its source. Requalification should be performed periodically to ensure ongoing compliance and consistency over time. Now, you might be wondering why qualification is so important. Well, it helps us ensure comparability with the reference standard, or RS, which is our benchmark. This reduces the need for frequent, costly requalification of RS itself, saving resources while maintaining quality. It's also critical to maintain analytical consistency across different batches and over time. From a regulatory perspective, the solution must be traceable back to pharmacopoeial standards, which are recognized benchmarks for quality and safety. Ultimately, these expectations help us ensure that our working solutions meet the highest standards of quality and traceability, aligning with regulatory requirements and supporting the integrity of our work. Let's walk through the qualification procedure overview. First, we start with the procurement of the reference standard, or RS. This is crucial because the RS serves as the benchmark for our testing. Next, we move on to the selection of a suitable working standard candidate, the WS. Choosing the right WS is vital to ensure consistency and reliability in our process. Once we have our WS candidate, we conduct testing against the RS. This step helps us verify that the WS aligns with the expected standards. Following testing, we perform a statistical evaluation. This involves analyzing the data to confirm the accuracy and reproducibility of our results. After validation, the final step is certification and approval, granting us confidence in our WS for ongoing use. Now, let's talk about how we select the material for the working standard. We use an approved batch of API or raw material, ensuring it meets our quality criteria. The key criteria include high purity, meaning the material must be above our specified limits, and it should come from a stable and representative batch. Ideally, we prefer to use the current production batch, as it best reflects the material we are working with in real-world conditions. All these steps help us maintain rigorous quality control, ensuring the reliability and accuracy of our testing processes throughout the entire qualification procedure. Let's take a look at the analytical tests we use for qualification when comparing our product with the reference substance, or RS. First, we start with a description of the material to ensure it matches what we expect. Identification is a crucial step, and techniques like IR, UV, or HPLC are commonly employed to confirm the identity of the compound. These methods help us verify that the substance is correctly identified and pure. Next, we perform an assay, which measures the active ingredient's concentration to ensure it meets the specified standards. We also check for related substances. These are impurities or degradation products that could impact the quality or safety of the product. Water content and limit of detection, or LOD, are also important parameters. They help us assess the purity and stability of the material by quantifying residual water and detecting trace impurities. A key point to remember is that IR spectra comparison is often a required part of this process. Comparing IR spectra provides a quick and reliable way to confirm the identity and purity of the substance, making it a valuable tool in our qualification tests. Overall, these analytical tests form the backbone of our quality assurance process, ensuring that our product consistently meets the necessary standards and is comparable to the reference standard. Now let's talk about the acceptance criteria for our results. Essentially, we expect our results to fall within predefined limits, which helps ensure consistency and quality. They should also be comparable to the reference standard, or RS, that we've established. When we say typical, we're referring to the expected variability, such as an assay within plus or minus 1 to 2%, based on our standard operating procedure, or SOP. Additionally, impurities must be within specified limits to meet quality standards. Moving on to the assay assignment, this involves determining the assay of the working standard, or WS, against the reference standard. We typically use analytical methods like HPLC, gas chromatography, or titration for this purpose. It's important to remember that we need to apply correction factors, especially for moisture content and impurities, to get accurate results. And one key point here is that the RS assigned value must always be used. We should never assume it to be 100% because that could lead to inaccuracies. So overall, maintaining these criteria and applying proper correction factors ensures our results are reliable, comparable, and within quality standards. Let's take a closer look at some of the key documentation requirements that are essential for ensuring quality and compliance. First, we have the qualification protocol and report. These documents provide a detailed plan and summary of the validation process, confirming that equipment and procedures meet the necessary standards. Next are the raw data and chromatograms, which are the fundamental records of the actual testing processes. They offer traceability and proof that tests were conducted properly and results are accurate. Then there's the certificate of analysis, or this is a critical document that summarizes the test results and confirms that a product meets specified quality criteria. It's often relied upon for regulatory and quality assurance purposes. Traceability records are also vital. They ensure that every batch or lot can be traced back through the production and testing history, which is crucial for any investigations or recalls. Moving on to labeling, there are several key elements to highlight. The lot number helps us identify specific batches, which is important for tracking and quality control. Potency information indicates the strength or concentration of the product, ensuring it meets the expected standards. Lastly, the expiry or retest date informs users about the product's shelf life and when it should be retested to maintain quality. All these documentation and labeling elements work together to uphold transparency, traceability, and confidence in our products, aligning perfectly with our overall goal of maintaining high standards in quality assurance. Let's talk about requalification and lifecycle management. It's essential to perform periodic requalification to ensure our systems stay in optimal condition. Typically, this is done annually or according to the procedures outlined in our SOPs. But there are specific situations when requalification becomes even more critical. For example, when a new RS lot is introduced, or when a working solution is nearing its expiry date, we need to requalify to confirm everything remains compliant and effective. Maintaining ongoing stability monitoring is a key part of this process. We need to keep a close eye on the stability of our products, making sure they continue to meet quality standards over time. Additionally, storage conditions play a vital role. They must be consistently monitored and maintained to prevent any degradation. Overall, these practices help us manage the product lifecycle effectively, ensuring safety, quality, and compliance throughout the process. So, to wrap up, I'd like to emphasize that WS qualification is absolutely critical for achieving GMP compliance. It's not just a checkbox, it's a fundamental part of ensuring that our processes meet the highest quality standards. We need to make sure that there is clear traceability to the reference standard, which means every step and every measurement can be tracked back accurately. This traceability is essential for accountability and consistency. Additionally, scientific justification plays a key role. We must have a solid scientific basis for our methods and decisions, which helps build confidence in our results and ensures regulatory acceptance. Robust documentation is also vital. Proper, thorough records support everything we do. They serve as evidence that our processes are controlled and compliant. All of these elements together support data integrity, which is the backbone of reliable analytical results. When data is trustworthy, we can confidently make decisions and satisfy regulatory authorities. In the end, these best practices help us produce consistent, high-quality products and maintain the trust of regulators and customers alike. For further support, please don't hesitate to reach out to us. We're here to help with any questions or additional information you might need. Remember, we're committed to ensuring you feel confident and well-informed about everything we've covered today. Thank you so much for your time and attention. We truly appreciate your engagement and look forward to assisting you further.